Fertilization is when a sperm penetrates an oocyte to form a zygote. Now, after ejaculation, the sperm has to move up the female genital tract to somewhere around the ampulla of the uterine. The spermatozoa can only fertilize the oocyte after capacitation and the acrosome reaction. So what is capacitation? Well, this is the conditioning of the sperm in the female reproductive tract where the glycoprotein coat and the seminal plasma proteins are removed. Morphologically, there is no visible difference. However, only capacitated sperm can actually pass through the corona radiata and fertilize the egg. Once this capacitated sperm passes freely through the corona radiata, you then have the acrosome reaction by binding of the sperm to the zona pellucida. Now, this is a very important step because the zona proteins induce the release of enzymes from the acrosome, allowing the sperm to penetrate the zona pellucida itself, and contact of the sperm head with the oocyte surface leads to what we call the zona reaction. Now, this zona reaction is extremely important. The reason for this is that the alteration in the zona pellucida surface glycoprotein, specifically the ZP3, prevents the penetration of additional sperm. So it basically says, all right, we're closed for business, all right? This is it. Once sperm's gotten through, we're not letting anyone else through. This prevents multiple sperm from entering into the ovum, or what we would call polyspermy. Now, once the sperm penetrates the oocyte, there is fusion of the oocyte and the sperm cell membranes. The oocyte then resumes the second meiotic division, and the formation of the second polar body happens at this point. Spermatozoon moves towards the female pronucleus and forms its own male pronucleus. The tail now degenerates. The two pronuclei then fuse. The combination of the 23 chromosomes in each of the pronucleus results in the zygote that has 46 chromosomes. The DNA replication occurs similar with normal mitotic division, and you will now have a common mitotic chromosome spindle or cleavage spindle. And zygote actually means yoked or joined. And so the female and the male has now joined and yoked together to form the zygote. So the result of fertilization is a zygote, and the first cleavage division of the zygote occurs to form the two-cell zygote. Now, cleavage itself is simply the increase in the number of cells without actually increasing the volume. And the zygote would contain two blastomeres. These blastomeres are totipotent cells. Totipotent. What does toti mean? It means all, okay? It means that up until the 8-cell stage, any one of these blastomeres can actually form a complete embryo on its own. And this stage is called the blastula. It's very important because it means that if these cells are separated, you would end up with identical twins, monozygotic twins, because they're formed from the same zygote. And they contain identical genetic material. Now, contrast this to dizygotic twins, or fraternal twins, that are formed from two different zygotes, and they would be as different from one another as any two brothers or sisters. Back to our embryo. The blastomeres continue to undergo rapid mitotic divisions, which leads to a further increase in cell number, again without increasing the volume, which, remember, is the process called cleavage. Now, as the cell count increases to 32, this forms a compact cell mass called the morula, and the morula has an inner cell mass and an outer cell mass. Conjoint twins or Siamese twins may occur if during the split of the embryoblast, the inner cell mass does not completely split apart. These twins are then joined by a tissue bridge either at the head, the thorax, or the pelvis. Now, the first recorded successful separation of conjoint twins was actually carried out by a Swiss surgeon, Johannes Fatio, in Switzerland, sometime around 1689, really long time ago. The patients were a pair of newborn girls called Elizabeth and Katharina, both of whom recovered fully from the surgery. So it is possible to separate conjoint twins, and it has been for quite a number of years, if they have developed individual organs. It's a lot more complicated when the twins share vital organs. Now, in 6 days, the embryo finally makes its way rolling down the fallopian tube into the uterine cavity. And we have entry of fluid that starts to form a cavity, and that's now a blastocyst. And the cavity itself is called a blastocyst cavity, with the inner cell mass forming the embryoblast and the outer cell mass forming the trophoblast. So let's summarize this first week. We have ovulation, which is oocytes from the ovary into the fibrilla of the ovarian tube or fallopian tube or uterine tube. You can pick whichever name you want to call it, all the same. Fertilization will happen between 12 to 24 hours thereafter ovulation in the ampulla of the ovarian tube or the uterine tube or the fallopian tube. And you will get to the 2-cell stage, zygote stage, about 30 hours after fertilization. You will have the morula, which remember is 32-cell stage, in about 2 days. Now the blastocyte, after 6 days of fertilization, is now at the point where it is ready for implantation in the uterus. Implantation occurs at the end of the first week. So the uterus is lined with endometrium, which is a specialized layer containing numerous glands and blood vessels that are ready to nourish the embryo. The blastocyte has differentiated into the embryoblast composed of epiblast, the hypoblast adjacent to the blastocyst cavity. The trophoblast is composed of the cytotrophoblast made up of single cells. These cells divide and contribute to the growth of the trophoblast itself. Now the syncytial trophoblast is a cytoplasmic mass with several nuclei, and continually the cytotrophoblastic cells fuse with the syncytial trophoblast and increase its size. The syncytial trophoblast grows into the endometrium and engulfs endometrial decidual cells that contain the glycogen and lipids that provide nutrients to the embryo. The syncytial trophoblast grows into the endometrium and also erodes blood vessels that form lacunae within its trophoblast. And this trophoblast blood vessels form the uteroplacental circulation. Syncytial trophoblast produces human chorion gonadotropin, HCG, which enters maternal circulation, maintaining corpus luteum. So estrogen and progesterone are needed to maintain the pregnancy. And this is what would be detected in the urine test when you're doing a pregnancy test. And with that, we've covered fertilization and implantation.